Hello, this is an introductory lecture for the teaching modules of the One Health Regional Network Horn project. This lecture forms part of the Tier 1 One Health Concepts curriculum program. My name is Professor Eric Fevre, and I work on the epidemiology of zoonotic diseases at the University of Liverpool and jointly with the International Livestock Research Institute in Nairobi in Kenya. The subject of this lecture is the ecology of infectious disease. Infection is the invasion of an organism's body tissues by disease-causing agents and the establishment of a population of those agents in a new host. Disease-causing agents might include bacteria, viruses, parasites, or fungi, and we shall look at some specific examples later in this lecture. Some infectious diseases are non-communicable. That is, they are not spread between hosts, but may be acquired by a host from the environment. For example, tetanus is an infectious disease acquired from contact with, for example, soil. A communicable disease is a disease that can be spread directly or indirectly from one host to another. Understanding the way in which hosts interact with each other and the way in which disease-causing organisms move between hosts is an essential element of understanding communicable disease biology. Zoonotic diseases are infectious diseases with animal reservoirs that can cause disease when transmitted to humans. An acute disease has a rapid onset and rapid reproduction of the disease-causing agent, leading to often short but possibly severe courses of disease with a large number of infectious agents produced in the individual host for onward transmission. This is a characteristic of many viral infections. A chronic disease has a slow onset of agent reproduction, which elicits a manageable response from the host and from the host's immune system. The infection period may be extended over long periods of time with slow rates of onward transmission. When an individual is exposed to a pathogen, there is a pathway followed for both infectiousness, the ability of those pathogens to infect further hosts, and disease, the impact of the infection on the host itself. This timeline will vary by pathogen and by host, and strongly influences how a pathogen spreads in a population. Let's look at the diagrams on the right-hand side of this slide to understand these different states. First, in the top part, let's look at infectiousness. That is, the ability of a host infected with a pathogen to pass on that pathogen to other hosts. First, the host starts as susceptible, then becomes infected through one or other ways in which that pathogen may invade that host. After infection, there is a latent period, a period in which the host is carrying that pathogen is reproducing in the host, but it's not yet at a point at which that host can transmit that pathogen on to other individuals. After the latent period, we move into the infectious period, where the host is shedding the pathogen and other hosts are able to acquire that pathogen from the host that is shedding it. That original host is now termed infectious and is a risk in terms of transmitting that pathogen on to further individuals. After the infectious period, we may move into a non-infectious period, where, for example, the host has dealt with the infection through their immune system, and while it may still be carrying some infectious organisms, is no longer transmitting those infectious organisms to further hosts. The second diagram underneath shows a very similar pattern, but it's the dynamics of disease, that is, the impact of the infection on the individual host rather than on the population at large. So a susceptible host becomes infected and then moves into an incubation period. And the incubation period is the period in which the host is carrying the pathogen and that pathogen is reproducing but not causing any disease. Then, roughly about the time that that pathogen becomes infectious, that host becomes infectious, the host moves into the symptomatic period, that is, shows symptoms of infection and is in a diseased state. And beyond that symptomatic period, the host moves into a non-disease state. Perhaps, for example, it has recovered from the infection. When a pathogen spreads in a population, it will do so according to some predictable rules specific to that pathogen and dependent on the individual level parameters related to that pathogen, such as the length of the infectious period, the length of the latent period, and so on, as defined on the previous slide. So, for example, one primary case will infect further individuals who are the secondary cases. Those secondary cases will incubate the infection and then transmit on to a third level of infections called the tertiary cases, and so on through until, in some cases, the entire population is infected. In this context, you may hear about R0, R0 pronounced R0, which is a mathematical term that indicates how contagious an infectious disease is. It is sometimes called the basic reproduction number because a spreading pathogen is reproducing itself as it spreads. And R0 essentially defines how many new infections arise on average from each infected host in a population. So in the image on this slide, on average, each infected host is resulting in 2 further infected individuals. So the R0 is on average 2 in that population, and the disease is spreading. When epidemiologists look at data showing the number of cases of a disease over time, they draw what we call an epidemic curve. On this slide, we see the epidemic curve for an outbreak of severe acute respiratory syndrome, or SARS, in Hong Kong in 2003. The epidemic curve essentially describes the course of an infection in a population over a period of time. How long an outbreak takes to get started after the first primary case, how long it takes to reach peak transmission, and how long the tail on the graph is, where the tail leads towards the last case, is all determined by the transmission parameters that we have described earlier. Understanding, given the parameters associated with a particular pathogen and the population which it affects, the shape of the epidemic curve is essential to understanding and predicting the course of infectious disease outbreaks. While we have seen that there are a number of biological factors relating to a pathogen and its host population that influence disease transmission, there are also another range of influences of how quickly transmission may occur. And it is essential to understand all of these different influences when trying to understand infectious diseases. So, for example, there are aspects related to the host itself. The age of the host, the sex of the host may influence transmission. Behavioral traits, how often people have contact with each other or individuals have contact with each other. The nutritional status of the host, the health status of the host, the immune status of the host, for example, whether hosts are vaccinated or not, will all influence whether or not an organism, a pathogen, is able to infect that host efficiently and spread from it. There are also factors related to the agent or the pathogen, its innate infectiousness, pathogenicity and virulence, the immunogenicity, its antigenic stability, and the persistence of that agent in the host or in the environment. And environmental parameters may also have a big influence. Environmental parameters may include climate, housing, geography, the occupational setting, the environmental quality, or food. All of these factors influence the rate at which an organism may spread through a population. The agents that cause infectious disease fall into 5 groups. Many members of each group are multi-host and have animal reservoirs, making them relevant when considering diseases in a One Health context. Diseases may be caused by viruses, and an example of a zoonotic virus is yellow fever virus or rabies virus. Diseases may be caused by bacteria. An example of a zoonotic bacterium is Coxiella burnetii, which causes Q fever, or Staphylococcus aureus. Diseases may be caused by fungi. An example of a One Health relevant fungus is blastomycosis. Diseases may be caused by protozoa. An example of a zoonotic protozoan is the trypanosome, which causes human African trypanosomiasis, or leishmania, which causes leishmaniasis. And diseases may be caused by helminths or worms. And an example of a zoonotic helminth is Taenia solium, which causes cysticercosis, or Echinococcus, which causes echinococcosis. Do use other resources to explore examples of viruses, bacteria, fungi, protozoa, and helminths that cause disease in animals and in people. There are many ways in which infectious agents. We have already mentioned zoonoses. Zoonoses are a subgroup of infectious pathogens, and zoonoses are diseases that are naturally transmitted between people and vertebrate animals, including wildlife, livestock, or domestic pets. Zoonoses, in fact, constitute about 60% of all human pathogens. And being zoonotic is an important strategy for that pathogen to infect multiple hosts, increasing the size of the environment in which that host can live and multiplying the transmission opportunities. Examples of zoonoses are rabies for a virus, bovine tuberculosis for a bacterium, cysticercosis for a helminth, and leishmaniasis for a protozoan. This series of Tier 1 lectures has a specific lecture on zoonoses, which I encourage you to also listen to. Another essential definition is that of a host and of a reservoir. A host is an organism capable of acquiring an infection and harboring the infectious organism. The organism may or may not thrive in that host organism. A reservoir plays a much more important biological role with respect to transmission. A reservoir host is one that either alone or together with other species plays a significant role in sustaining the pathogen with respect to transmission to a specific target population, because the host population is large enough to maintain that pathogen. In other words, a reservoir population provides a natural environment in which a host can thrive and multiply, ready to infect a specific target population of interest. For zoonotic diseases, the target population is usually a human, and the host population, host or reservoir population, is usually an animal. For many human diseases, which do not have animal reservoirs, other human beings are the reservoir for non-infected humans. So, for example, measles is an exclusively human disease, and infected humans are the reservoir for other non-infected humans. Controlling infectious diseases essentially involves preventing either transmission or infection with a pathogen within a population. As directly transmitted organisms spread from host to host in a population, control will often involve reducing transmission, that is, reducing the risk of a pathogen moving between individuals. For sexually or intimate contact transmission, this can involve preventing such contact. These kinds of interventions often involve social interventions, for example, giving advice on sexual abstinence, providing options for condom use, and so on. For droplet-type direct transmission, encouraging the use of a handkerchief as a very simple measure when sneezing may reduce the risk of transmission. When transmission cannot be stopped, interventions may be targeted at preventing infection of new hosts. Measles infection can be prevented by vaccinating susceptible hosts. If enough individuals in the population are vaccinated, around 80% in the case of measles, infection drops below a threshold where each infected person gives rise to less than one new case. As we've seen on the previous graph, this would be an R0 of less than 1, where each infected individual gives rise to less than one secondary case. and the agent can therefore no longer be sustained in that population. In the case of measles, recently an anti-vaccination movement in some parts of the world has led to an increase in the size of the susceptible population after many years of intensive childhood vaccination for that disease, and measles has resurged as a result. The transmission cycle of multi-host pathogens needs to be well understood to design effective control. Just like we saw with directly transmitted pathogens, control of multi-host organisms can be either through preventing the risk of transmission between members of the population or reducing the chance of infection in an individual. Both these approaches will reduce spread in a population. It may be more efficient or cost beneficial to target control at the non-target host. That is to say, to target control at the reservoir. For example, treat animals to prevent them from infecting humans, or control vectors to prevent vector-borne transmission between different host species. For example, vaccinating cattle against bovine TB reduces the size of the livestock reservoir of infection, or controlling tsetse flies or mosquito populations reduces the chance of those vectors biting infected hosts and transmitting infections to new uninfected hosts. Finally, I want to touch on the international health regulations and how they relate to infectious diseases. The International Health Regulations from 2005 are a legally binding instrument of international law applicable to all member states of the World Health Organization, which currently consists of 196 countries. And this helps countries work together to prevent and control disease in humans while having a minimal impact on travel and trade. In animal populations, the equivalent is the OIE phytosanitary regulations. The first formal international cooperation on disease control was the International Sanitary Regulations in 1851, resulting from European cholera outbreaks. The World Health Organization was constituted in 1948. In 1969, it adopted revised and consolidated international sanitary regulations, which were named the International Health Regulations of 1969. These were mainly concerned with cholera, plague, and yellow fever, and later smallpox. They were dependent on country-level notifications and did not include a mechanism for preventing the spread of many diseases. From 1995 to 2005, it was recognized that these needed to be updated, and during that period, they were under revision, resulting in today's International Health Regulations 2005, which came into force as a legal document in 2007. The World Health Organization states that the International Health Regulations help build countries' capacity to detect, assess, and report public health events, that the International Health Regulations include specific measures at ports, airports, and ground crossings to limit the spread of disease and health risks to neighboring countries, and involve a comprehensive global alert and response system so information is available and response operations are coordinated effectively. These regulations also allow the World Health Organization to declare public health emergencies of international concern, which are defined as extraordinary events determined to constitute a public health risk to other states through the international spread of disease, and which potentially require a coordinated international response. Since 2007, when these regulations came into force, the WHO Director General has declared 4 public health emergencies of international concern. First, in 2009, the influenza pandemic caused by influenza H1N1. In 2014, due to setbacks in polio eradication efforts. In 2014 also, the West Africa Ebola epidemic, and in 2016, the Zika virus outbreak. As I mentioned, the equivalent regulations touching on animal health are defined under the statutes of the OIE, the International Organization for Animal Health, and these similarly define special measures which may be taken by countries to prevent the spread of animal-related disease between countries. In summary, we have seen that infectious diseases can and should be studied given an ecological context. We must understand the ecological environment in which diseases are transmitted to understand how they spread and how we can control them. Understanding the way in which diseases move between hosts is an important part of understanding how they spread both between and within populations. The transmission of pathogens can involve complex and fascinating pathways. And the control of disease is largely about controlling transmission. So understanding transmission methods has very practical outcomes. On this final slide, you'll find some links to additional resources, references available in the open access literature. I encourage you to take aspects of the lecture that I have given to you and explore those elements of it that you find particularly interesting and search for additional references to expand your understanding of those topics. Thank you very much.